हेलो लर्नर्स इन दिस वीडियो हम जानेंगे Corynebacterium diphtheria के बारे में and at the end of this video you will be able to figure out why this cute guinea pig is crying, what is this whitish layer over the mouth, what is this structure, and what is this little beautiful thing. So let's begin the video with a statement. Diphtheria is toxemia and not bacteremia. You'll get this statement later in this video. So our objectives in this video would be to learn about some general properties about the Corynebacterium diphtheria, its virulence factor, the pathogenesis of the disease, clinical features, the laboratory diagnosis, and at last we'll be summarizing the whole video. So, starting with general properties. This Corynebacterium diphtheria is also known as Klebs loeffler bacillus. So it's obvious why it is known as Klebs loeffler bacillus. Klebs and Loeffler would have been some person, but they were not suffering from the disease caused by Corynebacterium diphtheria. Klebs discovered these bacteria, and Loeffler first cultured these bacteria, and that's why they are known as Klebs loeffler bacillus. And nextly, they're non-motile bacteria, they're aerobic bacteria, they're gram-positive bacteria, they're non-sporing bacteria, and they're non-capsulated bacteria. Remember this non-sporing thing because you might be confusing non-sporing thing with some other thing which you will know later in this video. Okay. Now, talking about the virulence factor about this bacteria, I'll again repeat diphtheria is toxemia and not bacteremia. Now, it's time to clear this up. Why I'm saying diphtheria is toxemia and not bacteremia? Because it is the diphtheria toxin which is produced by the bacteria, which is the main villain of the story and is responsible for the infection which is produced by the bacteria and not the bacteria itself. And this diphtheria toxin has got two important properties. Firstly, it has got toxicity, and secondly, it has got antigenicity. This toxicity is responsible for the infection produced by the bacteria, and this antigenicity is another property which is immensely harvested in the field of vaccination, which we'll be seeing later in this video. Now, this toxin has got some special affinity to heart and nerves, and thus producing cardiovascular and nervous manifestation too in the disease known as diphtheria. So, this bacteria, Corynebacterium diphtheria, produces a toxin which is a polypeptide chain, has got A and B fragments. This toxin, the B fragment of the toxin helps the toxin to attach itself to the cell, and then the A fragment internalizes itself, and this A fragment then inhibits the protein synthesis inside the cell, and this inhibition of protein synthesis then kills the cell. Right? Now, this bacteria can manifest itself into various ways. So firstly, the most common manifestation is in the form of respiratory diphtheria, wherein it can affect the facial or the pharynx region, or the nasal region, and the most fatal region is the larynx the larynx region. Secondly, it can also manifest itself as the cutaneous diphtheria, wherein it forms ulcers like this. And lastly, it can manifest itself as systemic diphtheria, wherein it affects the nerves, causing palatal paralysis like this. Here you can see this part of the upper soft palate is drooping down just because the nerve supplying this palate has got paralyzed due to demyelination process caused by this bacteria. And earlier we said that it has got some special affinity to heart too, so it also causes myocarditis, right? So these are the manifestations produced by Corynebacterium diphtheria in diphtheria disease, right? Nextly, let's see the proper pathogenesis, how this toxin causes the production of this respiratory diphtheria manifestation, right? So this toxin damages and necroses the epithelial cells of the tonsils and pharynx and forms exudate and ulcer. And these ulcers are surrounded by a dirty whitish layer of pseudomembrane. This dirty whitish layer over the tonsil and the pharynx is known as pseudomembrane because it bleeds on removal. It is called as pseudomembrane because it bleeds on removal. If we try to remove it, it will not remove easily, and it will bleed on removal, right? And the pseudomembrane, if someone asks you the content, you can easily say it contains neutrophils, it contains RBCs, and it contains bacteria, right? Now, the next most fatal thing about the pseudomembrane is that if it extends into the nose and larynx, it can obstruct the airway pathway and can cause fatality by asphyxial death. So, this is a medical emergency and needs an early intervention in order to prevent the child from succumbing to the asphyxia. Right. Now we come to the most important part of this video, the laboratory diagnosis of Corynebacterium diphtheria. Now this diagnosis can be made on two broad bases. Either we can demonstrate the bacteria or we can demonstrate the toxin. In demonstration of any microbe in microbiology, what we do first, we first collect the sample, we perform microscopy over it, we then culture it, we perform some biochemical identification test, we then do serology, molecular test, and all. So in this bacteria, the sample collected is the throat swab or the piece of pseudomembrane which is formed over the tonsil and the pharynx. Nextly, we perform microscopy using two stains, the Gram stains and one special stain known as the Albert stain. And nextly, we perform the culture, and a little and of little importance is the biochemical identification test. Nextly, in the demonstration of toxin, we perform some in vivo no, we don't perform in vivo. It was earlier used, but it is not used now. So we used to perform in vivo demonstration technique of toxin in Winnipeg, and in vitro techniques like LX gel precipitation test and molecular like PCR analyzer or immunochromatography. We'll be seeing this in more detail. So once you've got the throat swab as the sample in a laboratory, so you have to perform microscopy. So we first perform the Gram stain over the sample. So in this Gram stain, as you can see, that's a very beautiful Gram stain where you can see this bacteria with these ends like bubble ends. This bubbling is known as the clubbing, and club is the Greek word for coryne, and that's why it is known as Corynebacterium diphtheria, right? So it's a Gram positive, staining positively and showing this club shape. Nextly, we've performed the special stain, which is the which is the Albert stain. Albert stain, as you can see, it characteristically stains the bacteria green, and at either of the ends you can see these bluish to blackish colored granules. Now these are not spores, remember, these are not spores, but these are metachromatic granules, which are just storage granules. And now you can here see that these bacilli under Albert stain are arranged in very randomized fashion, like just like this. So what language can you relate it to? It is looking like Chinese, right? So it is nothing but the Chinese letter arrangement, we also call it as. So this is all about microscopy. Nextly, we come to culture. In culture, we have got two main cultures. The first one is the low floor serum slope, wherein the bacteria form small creamy colonies, and nextly we have got potassium tellurite agar, wherein the bacteria forms black colonies. These black colonies are due to the accumulation of reduced tellurium in the agar, right? Now nextly we come to the biochemical identification test, wherein we have got these some important tests like catalase, oxidase, indole, citrate utilization test, and urease negative. It is not important to remember all these, but you can remember this urease negative as one of the characteristic property which can be asked in MCQ. And some outstanding points here in these biochemical identification tests are this: his serum sugar media we use, wherein the bacteria ferments glucose, and nextly it is pyrazinamidase negative. Now don't go very deep into the detail because it is not for undergraduation level. You just remember it can be asked as an MCQ question, right? Now let's come to the demonstration of toxin. So the toxin can be demonstrated as also stated earlier by in vitro and in vivo method. In vivo method, we used to do we used to use this cute little guinea pig. Now this little guinea pig is crying. Guinea pig, hum to me use nahi karne wale, so goodbye for now, because we have got some advanced technique wherein we take horse serum agar in the form in a petri dish, and we put an antitoxin impregnated filter paper strip, and then we take colonies from our cultured colonies, and then we make streaks on uh, this agar, and we label them as 1 2 3 4 5, and then we wait for some time, 24 to 48 hours we wait for them. Now what happens that this antitoxin strip starts releasing antitoxin, and these streaks of bacteria uh, will produce toxin. Now when this antitoxin and toxin meet together, they form they form something called as precipitation line, and the pattern of precipitation line tells us what type of strain the bacteria is. And this special method of demonstrating toxin in Corynebacterium diphtheria is known as Alex gel precipitation test. Now here this precipitation test is a process which is discussed in some other video, wherein we discuss many videos of antigen antibody reaction. You can check it out later. Currently, just for now, just remember that antitoxin and toxin, they combine with each other to form visible precipitation line. Now based upon this formation, these lines will be formed only when these bacteria will produce toxin. Now in this fifth streak line, can you see any such pattern of line or the precipitation line? No. That's why 1 to 4 are toxigenic, means 1 to 4 streak lines the bacteria are toxigenic, means they are producing toxin. That is why they're producing these precipitation line, while the fifth streak is the non-toxigenic. Now what is this pattern of this precipitation line? So the first pattern wherein the two cross each other signifies that the 1 and 2 species or the 1 and 2 strains are unrelated strains. The second pattern wherein there is a spur formation, it signifies that this strain 2 and 3 are partially related strain. And lastly, the pattern which is fused pattern signifies that 3 and 4 are identical strains, right? So this was all about Alex gel precipitation test. Nextly, I would like to shed some light upon this concept of lysogenic conversion, wherein initially the Corynebacterium diphtheria is a non-toxigenic happy bacteria with no toxicity. But what later happens, a bacteriophage comes, especially known as Corynenophage, which has got an evil genetic material. This bacteriophage then injects this genetic material into this non-toxigenic strain of Corynebacterium diphtheria, and once this genetic material gets included into the normal or the normal genetic material of the non-toxigenic bacteria, it converts the non-toxigenic bacteria into the toxigenic bacteria, and this conversion is known as lysogenic conversion. And once this toxigenic bacteria is formed, this then reproduces to form many toxigenic bacteria, and these all toxigenic bacteria produce the toxin which is responsible for the manifestations, right? Now some outstanding points to note down here is about the toxin is a special strain known as the Park William 8 strain of the diphtheria toxin has got the very good antigenicity, and this antigenicity, as also earlier stated, is immensely harvested in the field of vaccination, because antigenicity is the because when this toxin is inoculated or injected into humans, it causes production of antibodies against these toxin. Now in case of any future infection by the same bacteria, our body will already be ready with the antibodies, and it will be fighting against the bacteria, and thus preventing the disease to manifest itself. And this type of immunity is known as active immunity, right? Now in India, we give this strain of toxin in the form of toxide, which is included in the pentavalent vaccine, which is one of the parts of our national immunization schedule. So a question for you, a quick question for you. At what time do we administer this pentavalent vaccine? So you guessed it right. We administer it at 6th, 10th, and 14 weeks after the birth, right? Second point here to note is that antibiotics are ineffective in the diphtheria infection just because toxin is the main villain of the story. So antibiotics will not be effective against toxin, but except for skin lesions where bacteria play a important role over toxin, antibiotics can be effective in case of skin lesions. And lastly, we have got some commensal Corynebacterium too, which are known as diphtheroids. So in cases of sample collection and in cases of microscopy, these commensal bacteria could be uh, could confuse us with the pathogenic bacteria, right? Now, at the last, we need to summarize this video. So, Klebs Loeffler bacillus causes diphtheria. Diphtheria toxin is toxic and antigenic. This property is harvested immensely in the field of vaccination. The pseudomembrane characterizes the disease. Special stain, the Albert stain, stains is at green with metachromatic granules. Culture we use here are the Loeffler serum agar and the potassium telluride agar. The toxin is demonstrated by Elex gel precipitation test. And at last, antitoxin is immediately administered if diphtheria is suspected, because in case of any delay, it can cause airway obstruction and lead to fatality of the child. So it's an emergency. Thank you for watching the video. Like, share, and subscribe so that MindNotTired channel flourishes in future. Meet you in the next video. Till then, keep your mind not tired.